welcome everybody to this H3D Foundation webinar. You know how much I enjoy hosting these. And today we've got a special guest, Cecile Vernochet, and I'm going to introduce her now. Cecile Vernochet, PhD, is a discovery project lead at Gates Medical Research Institute, a role she's held since 2020. She focuses on supporting scientific discoveries, program development, and clinical trials for probiotic nutritional supplements in low and middle income countries, and advising on women's health product initiatives for the Gates Foundation. With over 20 years of experience in biological discovery research, Cecile's preclinical expertise spans stem cell biology to complex animal models for metabolic diseases, such as obesity, type 2 diabetes, and NAFLD-NASH. In the pharmaceutical field, she led a target discovery and validation laboratory at Pfizer, managing projects from preclinical stages through phase 2B trials. Cecile earned her bachelor's in biochemistry and molecular biology and a PhD in physiology from the University of Science, Nice, France. Her PhD research was supported by the French National Agency Against HIV. She extended her research expertise by completing a postdoctoral fellowship at BUMC and Harvard-affiliated Joslin Diabetes Center in Boston, USA. Cecile, we're so grateful that you're making time for us. I'm going to give you as much time as we possibly have, and so I'm going to hand over straight away to you after having said that there'll be a Q&A at the end. So please write down your questions, populate the chat, and I'll open the microphones at the end of the presentation. Over to you, Cecile.
Thank you, Nicola, for the introduction. And I just want to say that I've been part of the H3D program for a couple of years now and really enjoy this group. So thanks again for the opportunity to be there, and looking forward to have a conversation around the probiotic and LBP field. So without further ado, let's move on. So what I want to take you on a different maybe discovery journey that you might be used to or not really used to, depending on the field that you come from. And it's something that I really discovered in the past 5 years working for the Gates Medical Research Institute. So for people that are not aware who we are a subsidiary of the Gates Foundation. We support some of their program, clinical development, and really product development for market access. So we do anything from preclinical to small or larger clinical trials, and support all CMC and other aspects of the product development for some of the project of the foundation. So what the topic of today is really showing you a little bit of case, two very pragmatic case, one on a probiotic and one on a live biotherapeutic product, one to support infant and women health. So very briefly, you know, as you're aware, microbiome is highly related to health. In our human body, you know, our cells are not the majority of the cells. The Human Microbiome Project says the human body has, you know, hundreds of trillions of microscopic life forms living in it. So we live in a symbiotic aspect with good and bad bacteria in our body. And on the right, what you can see is the microbiome composition is different through the different part of our body or interface with the exterior. The composition of respiratory, oral, gut, skin, or vagina in that case could be very different in composition at time. It's also evolved and very different, you know, when you're a baby versus an adult, where with elderly people, and also it's influenced by diet and so many aspects of our life. So it's a very complex system, it's a very complex environment, but highly adaptable to any other influences, exterior and interior of our body. So as over the years we learn how microbiome and its composition influence health, we also learn how it can impact the disease progression. For some example, on the left, it's implicated or accelerate the progression of some of the heart disease, inflammatory bowel disease, liver disease, chronic kidney, brain, diabetes, respiratory cancer, and so on. But for today's topic, I want to take you through the journey of two special cases. Is a probiotic case of food dietary supplement that is called Bifidobacterium B. infantis to support weight gain in underweight infant in LMIC, where we've used at GetSMRI a product commercially available. And I walk through you the preclinical step that we took to support that clinical trial evidence. And then the second case is a live biotherapeutic that is now considered a pharmaceutical drug because it supports and claims to reduce bacterial vaginosis recurrence, an indication that is a disease. So the pathway for the LBP is a pharmaceutical drug. And this is really an integrated program where we started from scratch with the Vaginal Microbiome Research Consortium that is supported by the foundation. And we started from scratch by the candidate selection, and it's a preclinical aspect that I'm going to briefly describe. All right, so for the probiotic system, the probiotic and LBP share some commonalities. So they both, the live biotherapeutic and probiotic, they both contain live microorganisms such as bacteria, yeast. They compare to some chemical drugs and biological products, and they have really generally a lower adverse reaction rate and a good safety profile because they're largely commensal bacteria. They're not modified. They have been isolated from human, from food, from animals and environment. So they're generally considered as safe. They do have, though, because they're live microorganism, stability issues. They can be influenced by multiple factors such as dosage, transport, storage condition, and administration methods. And, as you imagine, if you end up with a dead good bacteria in a product, that might affect the efficacy that is looking after for that indication. There's many differences, and in that slide, I don't mean to be completely stating all the differences between the two, but just giving you a few examples. For the probiotic, for instance, even though it's a live bacteria, it's marketed as a food supplement or dietary supplement. And it's really, if you read the label, it supports health, which can be very general statement and hard to describe or to prove without, rigorous clinical trial. But they follow the manufacturing standard of the food and dietary supplement, which we know could be very different than a pharmaceutical of a drug aspect. The production cost is pretty low, and the clinical trials requirements are pretty none, really, or one or two, because it's considered safe and found generally in other humans. The LBP on the right, it's really applicable to prevention, treatment of cure of a disease or a condition in human beings. So it's a drug regulatory path. So for example, in the US, it would follow the FDA, or in South Africa, you would have to go through SAFRA. It has a high manufacturing cost because as a pharmaceutical, it has to follow the pharmaceutical good manufacturing practices, GMP. So then it leads to higher production cost and control for all good reason for safety. And the clinical trials are definitely mandatory because you have to prove safety and efficacy for an indication. So those are the commonalities, even though we still talk about the good bacteria implementation, but also critical differences that depend on the claim and the purpose of what company are trying to sell it for has a very different implication on a regulatory CMC and marketed aspect. So I wanted to give you an example of a probiotic, Bifidobacterium infantis, and it's a single strain product. And we've investigated in a clinical trial recently, and the result will come out hopefully soon this year in a publication to support the weight gain in underweight infant in low and middle income country. So single strain food dietary supplement path, an already existing product. So the unmet medical need is obviously huge. The undernutrition in infant contributes to 45% of death for children under 5 years of age. And then the wasting, affect 45 million of children, most live in Southeast Asia and Africa. So finding a way to support infant at a young age, nutritional recovery or status is critical. So in 2023, there was new WHO guidelines, and I'm just stating a few key takeaways for the management of infant under 6 months of age. It's really, there's nothing much that is stated here. It includes the, guidelines to support the mother, caretaker for physical and mental health. It's obviously, recommended for inpatient admission and some outpatient management guidelines. And really the prioritization is, supporting exclusive breastfeeding with consideration for supplemental milk if there is a severe undernutrition status. So the guidelines, so there's no really food supplement or anything in those guidelines that would address the delivering of nutrients, but and do not address the imbalance in the gut critical for digesting and absorbing nutrients, which is a critical need for this particular population that is highly vulnerable under 6 months of age. So I'm showing you, some type of mechanism, how, a probiotic is known in the literature, at least preclinically, and some clinical evidence as well of how a probiotic could support gut health. It is known to impact intestinal inflammation. It is known, obviously, to help balance and to benefit a balance toward healthful gut microorganism and, combat gut dysbiosis with pathogenic bacteria. And then ultimately, it's also been demonstrated at least in vitro, to repair intestinal barrier. So you can see how all of these components, if it works, that the probiotic in vivo could support infant underweight management by helping some of the key issues that they experience at a young age with infection and other pathology. So I listed here 3 modes of action: the gut microorganism balance, the intestinal inflammation, and intestinal barrier, because this is something that we can focus in vitro, and I'll show you an example in a little bit, preclinically, to investigate B. infantis efficacy. So there's no shortage of product around the world that contain B. infantis, and it's also for adult product as well. But it's really B. infantis is tailored to infant phase because it has the capacity to absorb and process human milk oligosaccharide that is present during breastfeeding and during lactation. So it makes it a very good probiotic combo at such a young age, and it's naturally found in infant, but really decrease when infants are challenged with infection, and is found at a lower level or absent in some of the population that are underweight. About 1 infant out of 2 do not have B. infantis anymore due to the challenge that they're experiencing. But it's naturally found at a high level in healthy babies. So there have been several marketed products. Here I show you example that you can find in high-income country or LMICs as well. They are found as a single strain. This is the one on the left, Vivo, the two, one on the left, Vivo, and the Vitimatic. It can be found in formulated powder for the baby formula. That's an example sold by Nestle in the middle. It can be found in combination of other probiotic to support, as they claim, better infant gut health. This is a Lallemande product. With a baby picture here, and then it can be found in adult product as well, because with multiple other combination of lactobacilli and bifidobacterium to support digestive system. So there is no one product, and it's a very different. It can come in very different formulation. So when Getsemerai started to be involved in that program to take it to the next stage of another clinical trial, the foundation had supported already a study called Synergy that was led at ICDDRB in Dhaka, Bangladesh. And that Synergy study investigated the product Evivo, which is a single strain B. infantis, and they administered it for 28 days in infants that were 2 to 6 months. They were severely underweight. And when they look at the colonization in that product in infants, and also look at the impact on weight gain that you can see in the graph in the middle, was spelled out for weight for age, this score. They saw that at the study completion compared to placebo, EVC001 single strain or the single strain plus an HMO to potentially support its colonization, that when was given to infant underweight, the infant underweight gained more weight during that period compared to placebo. So that was pretty exciting findings in a relatively small study. So the foundation asked us to take on that project and answer the question, can other commercial B. infantis product, not Evivo, but something else on the market, do the similar impact on weight gain? And because when we think about a recommendation or potentially supporting a new WHO recommendation, you have to understand if it's product specific or if it's strain specific. So if you can find any product around the planet that has a B. infantis, could that do the same job for weight gain? So we ran a constellation study that was run internally but occurred in Pakistan. We use another single strain called BI26 for another independent manufacturer, IFF. And we had a placebo control, obviously, and we mimic as much as possible the setting from Synergy that was given once daily for 28 days, and infants were between 1 to 4 months age. And the endpoint, our primary endpoint, are weight gain. So I would have loved to show you some results today. Unfortunately, they will come up pretty soon. So happy to share it at a later stage or send the publication when it comes out. So as you can imagine, we cannot test all the B. infantis product clinically around the planet. That would be a huge cost and really time constraint. So in addition to clinical trials, what else can we do to understand if the different B. infantis strains are equivalent in terms of their efficacy? So we have defined an in vitro, multiple actually in vitro models, and established some in vitro efficacy with a reference B. infantis strain. And then we compare multiple B. infantis commercial strains mode of action in vitro. And the ultimate question that we're asking here is, are B. infantis strains functionally interchangeable? So so the way that we established those two systems is one was an infant colonic simulator where we look at the probiotic candidate and the host microbiome interaction. And this is to investigate the mode of action of shifting gut dysbiosis or dominating the gut as it's supposed to be in a healthy infant status. And then the preclinical readouts are growth fitness. We know short-chain fatty acid secretion and production, lactate production, microbiome composition, and fecal pH are all important markers. And we collaborate with a company in Belgium, ProDigest, with a system, Baby Chime. I won't have time to go through that, but want to show you an example of how you can put together a preclinical package for comparison on different mode of action. And then the data that I'll show you today are on using a gut-on-a-chip system. We partner with a French non-for-profit microbiome biotech called Bioaster. And this is to really investigate the commercial B. infantis strains and the host interaction in a gut, in a miniaturized mid-throughput in vitro model such as the gut-on-a-chip. And that allowed two things. We added human-derived peripheral blood mononuclear cells, PBMCs, so we can address the impact of the B. infantis on immunity and immune cells polarization function. And within the same system, because of the monolayer of human intestinal cells that create a barrier, you know, you could investigate the capacity of the B. infantis candidate to impact the intestinal permeability. And obviously here we use trigger pro-inflammatory stimuli, LPS, to create a barrier crash and decrease its integrity. And then we investigate how B. infantis, if B. infantis can repair it, and how the different B. infantis are they similar in terms of efficacy, or some of them are none or greater efficacy. So, so we look at cytokines as a readout. We looked at the TIR, the intestinal barrier integrity, which is electric resistance to a system. And we also looked at transcriptomic, you know, what type of mechanism, if there is an effect, what type of genes are responding to B. infantis candidates. So the gut-on-a-chip, if you're not too familiar, we use a commercial product called Mimetas. That has a lumen channel and a blood channel, and in between a collagen and in a lumen channel, you add the cells and then create over time a barrier between the lumen and the blood supported by the collagen. And then you can add components in either the blood component, where we're going to put the human PBMCs on, and look at the response of the intestinal cells in the lumen channel, and where we also had the B. infantis condition media. And then to create inflammatory damage and storm, we had liposaccharide LPS, so that elevate the PBMC secretion in very pro-inflammatory cytokines. The cytokines damage the epithelial barrier, and then the resistance of the intestinal barrier drop. And then when we add the conditioned media of bacteria, we can see how the TIR is repaired. So started on the left, we're gonna look at the inflammatory response. We know IL-8 is a cytokine that is not desired to be activated, and as expected, the LPS, the red bar, is elevating IL-8 compared to control in the white bar on the left panel. And what you can see is we use the ATCC 15697, which is a B. infantis reference strain, and additional other B. infantis commercial product, BI26, CVC001, Bifino2, and others. and a combination as well. And when you see that all the commercial product and the reference strains B. infantis were capable of reducing the islate secretion from the intestinal cells. So there is a statistic significant here and a reduction on islate, but it was not able to block it all. But all the key question here are all the B. infantis doing similar impact? And the answer is yes, compared to the reference B. infantis strain. On the right, we have the third mode of action. Does that repair, protect the intestinal barrier? So what you see in the right is the baseline. What you see in the red is the LPS capacity to decrease, like deteriorate the tear and the integrity. And then in blue, yellow, pink, green, and overs, you see the addition of the conditioned media of the bacteria. And all of them were able to improve the tear, so repairing, protecting the intestinal barrier against the pro-inflammatory challenge. Are one better than others? If you had to pick, maybe BI26 is the less potent, and BFINO2 and EVC, the other one, are slightly more potent, but at this stage, they're all within the same range and doing a similar impact. So this is an example how we can, are they interchangeable, at least in vitro, on two key modes of action? We're going to say yes to that so far. We also investigate their growth in presence of different substrate, and in our hand, they show similar growth impact. And they're all efficacious for the tear protection effect, and they all decrease the cytokine production of islate. So I'm going to quickly finish the case one and move to the case two with thanking all our partner, the company Biotherapeutic, Clinical Microbiomics, and the supplier for the HMO. We worked very closely with manufacturers in the probiotic field that were partner with us to that discovery work. So I want to thank him for their generosity and really collaborative spirit, and also all the Gestemera B. infantis team and other academic collaborator. All right, so moving on, I want to talk about a project that is now totally different. It's for women health, and this is now a good bacteria and multiple good bacteria actually that started from the discovery, from the candidate to the manufacturing to the clinical trial that has now completed, and again will be read out sometime this year. So really a pretty exciting time from the discovery in 4 years to the manufacturing to the clinical trial. So it's a very fast product development here that we are facing, and mainly because they are commensal bacteria and regarded as safe. So the package for submission in regulatory is pretty straightforward. So I want to call out the name of the clinical trial, Vibrant, which stands for Vaginal Life Biotherapeutic Randomized Trials. And this is led by Disebo Potlan at SAPFRA in South Africa, and Caroline Mitchell at MGH. But it's really, it takes a village and a large vaginal microbiome research consortia with many groups that have participated that I'll show you at the end. So there's a significant burden of bacterial vaginosis that affect women around the world. The general population prevalence of bacterial vaginosis is around 1 woman out of 4 or 5 across global geographies, but there's also marked heterogeneity within population. In the US, there's definitely a different ratio affecting African ancestral and Black population, Hispanic at 31%, and less in Asian, for instance, at 11%. And around the world, within the same continent, there's also large disparities of prevalence of bacterial vaginosis in women. In addition to be pretty, I would say, annoying, that I will describe how. It is also associated with increased risk of preterm birth, acquisition of sexual transmitted infection, HIV, and progression of HPV infection to cervical cancer. So there's many other reasons on why we should target and ameliorate the prevalence and recurrence of BV in population. And BV standard of care, according to CDC and around the world, is a treatment by antibiotic therapy. But unfortunately, it might cure your symptoms, but it doesn't stop the recurrence. And one of the two women that had BV once will have it again and again and again. And every time the therapy is antibiotics, which we know is not ideal and can lead for long-term issues about microbiome composition. The annual global economic burden of BV is around 5 billion, and that costs triple if you think about all the BV-associated PTB and HIV acquisition when included. So this is not a trivial number. So how does that work? So the natural vaginal health microbiome composition should be dominated by lactobacilli, ideally lactobacilli crispatus. We know that they produce H2O2, they lower the pH of vaginal secretion, and a series of events that led to a low inflammation status in that environment and other beneficial effect into that environment and disease. So this is actually a very simple microbiome. Good is one or two strain at a very dominant level, very different than gut. When the woman can potentially transition, they transition to another lactobacilli inners. There's not really BV associated with that, but we know it's not really looking good when you transition to that orange state. And then when you turn to have a very diverse vaginal microbiome dysbiosis with pathogenic bacteria, then that led to an increase in anaerobic bacteria, proteolytic enzyme, and then a creation of amine and proteolytic product, increased vaginal discharge and malodor, and the pH rise, and so on. So pretty detrimental to woman health and status. So antibiotics was the solution as a standard of care, but it stayed the same for over 50 years or 60 years and continued with no new discovery. And we know the problem. It's a quick fix, but it's not a long-term fix. And then obviously with microbial resistance and so on, you can see how it's not a long-term beneficial aspect for women health, and it really doesn't impact the recurrence. So women who had it once will have it on and on and on. So the critical challenge for that is that there is no animal model that model human biology. Humans are the only species with lactobacilli dominant vaginal microbiome. So when you think about therapy, it's hard to think how preclinical animal model can help here to mimic or investigate efficacy and to be a translation model. So if you can, you go straight to human with your product. And after antibiotic treatment, very few people reattain the L. crispatus dominance after the antibiotic clean the bad bacteria, I would say. And most have L. inner. So when you see that cycle, if you don't replenish with the L. crispatus dominant, you're not really fixing the problem. And we think that's why recurrence happens. So the solution and the hypothesis here is a vaginally applied Lactobacilli crispatus, LBP, can restore L. crispatus dominance and reduce the recurrence of BV following the standard of care. So replenishing after antibiotics to fix the recurrence. So Vibrant is a clinical trial. The group designed 2 LBP that are comprised of L. crispatus isolates, and they were derived from people from the US, but also from South Africa. We think that the potential geographic disparities of where the strains are coming from make maybe the product more capable of colonizing women around the world in a similar manner. But that's an hypothesis yet to be proven, that the geographic matters for where the strain comes from. And the 2 specific LBPs are consortia. So we don't give 1 L. crispatus, we give 6, or we give 15 strains. And how do we choose that is the candidate selection that I'm going to briefly walk through. But briefly, they were chosen based on bioinformatic algorithm that choose the best combination of the strain that best recapitulate the gene present in stable communities. And I'll walk through the process now. So there was a large effort in discovery to follow women that had a stable L. crispatus status over time. What you see on the left is individuals, 3 individuals here, and you see the composition in red where they had L. crispatus dominant, and you see that it followed the time of day. So longitudinal assessment of vaginal microbiome show L. crispatus dominant stable over time in those women. The Jacques Ravel group perform metagenomic and metatranscriptomic analysis of the L. crispatus dominant consortia and the presence of the gene, what makes the healthy vaginal microbiome gene content and its composition. And then in parallel, they derive individual L. crispatus from this community and identify 15 genetically unique L. crispatus from this community, from this woman with stable communities. So you're comparing a metagenomic, and then you also have individual strains that are coming from these communities. So what you see on the left is a representation on how many L. crispatus genes I found in L. crispatus dominated metagenomes of women from South Africa and US. You have about 3,300-ish gene represented in yellow in metagenomes. And then on the left in gray is the L. crispatus isolates. Now the single strain isolates genomes, and you can see it represents about 2,000 genes. So within the isolated strain, when you put together metagenome, it didn't represent the total metagenomic presence of the genes. And then they issue some type of barcode, and they look at how the strains and how many strains you will need to put together to resemble the maximal metagenome coverage. So when you look at the average coverage of metagenomes, you see that we peak at 75% of representation of the total metagenomes, and the number that gives you the minimal number of strains that you would have to put together to cover the maximum amount of genomes is around 6 strains. So this is the number that we're going to follow to put together the consortia in the next slide. So how are you going to choose these 6 strains that going to resemble the maximal metagenome coverage? So use a heuristic algorithm, and they're pretty much create a different combo of 6 strain of the 50 strain and see which one represents the most coverage. And we called these 6 strain consortia LC1 to LC6. And the way that we ended up choosing the best consortia candidate for moving forward to the clinic was looking at their capacity and comparing their capacity to produce an antimicrobial effect against BV-associated microbes from US and South Africa. And we chose obviously the best consortia to move forward based on this in vitro activity and antimicrobial activity. So this is how we picked the one consortia with 6 strains, and it ended up being free from women from the US and free from women from South Africa. So we are pretty happy that that discovery led to representation of both continents. And then the additional consortia that we move forward for manufacturing and is clinically investigated is actually the total of the 15 strain. And the hypothesis behind it is like, you know, women would have a hard time maybe to colonize with one single strain, but maybe if you propose 15, women around the globe would have a chance to colonize. Maybe one of the strain will be better in one woman versus another. So we're just trying to optimize the colonization by giving it more chance, but an hypothesis yet to be proven. So really briefly, as mentioned, it takes a village. And when we finished the candidate selection, we worked with a French company for manufacturing the tablets that will be vaginally inserted. So, and then for the clinical side, we wanted to know if that product, the same product, will have the same colonization in the US and in South Africa. In the US, that was led by MGH, Caroline Mitchell. And in South Africa, Isabel Portland and her team have finished to run the clinical trial. So same clinical trial, 2 sites, 2 tablet formulation, and the endpoint is looking at colonization. The randomized trial is as followed, and I'll finish very shortly to leave time for question. So we have a placebo arm. A woman comes with BV. They're treated with antibiotics as a standard of care. And then we looked at 3 different arms that has a consortia LC106, the 6 strain consortia. Either followed the antibiotics treatment, either for 7 days or 3 days, daily administered or overlapping with the antibiotics. And then we also interrogated, if you put more strains, LC115, all the 15 in one consortia, will you have a better chance of colonizing most women compared to LC106? So the trial has been successfully run in the 2 countries, and the analysis of the composition of vaginal microbiome is being done as we speak. So very exciting time in 4 years to see the product from thinking to manufacturing to clinical trials. So I mentioned it take a village, because between, the translation, the longitudinal, the discovery aspect of what a healthy vaginal microbiome should look like around the globe, there is a very large academic group that contributes to that, plus manufacturing and clinical trials. So I want to highlight the Caprisa team, I want to highlight the MGH team and all the discovery lab that participated to that effort. So with that further ado, I think I will take on any question as we speak. And I haven't been monitoring the chat, so I apologize for that in advance. Thank you for your time, and I'm good. Nicola?
Hi, Cecile. Thank you so much. Yeah, what an extraordinary. So 2 case studies that we for us to respond to. Really, really fascinating work. And I've unmuted everybody. So happy for people to raise hands with questions or to put their questions into the chat. I don't think we've had any come up so far, lots of AI, but no questions as of yet in the chat. I mean, it's extraordinary to have such a short time frame of 4 years. Where to from now? What's the next big leap for either of these projects, or what are you working on next, Cecile?
Yeah, so for the vaginal microbiome aspect, we are waiting on the result if those two hypotheses, like the geographic matter, the more than one strain will help more women, and will that product will colonize successfully in women. So if successful, there is a possibility to look at efficacy after looking at safety and colonization, looking at the efficacy of that product as is in a phase 2 for recurrence BV. If it's not enough, there is the opportunity to add other candidates or investigate other formulation component to add to the feed component. There is a large set of formulation and excipient or active ingredients that we could propose to support the colonization of these strains. So we're in a very interesting time of what are the data looking like, with a series of other ways to mitigate if that was not enough.
Thank you. I think it's Dina. Dr. Quinten, over to you.
Hi, yes, thanks, Nicola. Thank you for the interesting talk, Cecile. I'm curious, more from a point of interest, with the first study with the oral bioavailable probiotic, what is the concern with the stomach pH, the very low stomach pH, in killing of bacteria before it actually reaches the digestive tract?
Oh, you mean the impact of the digesting tract on the survival of the strains?
Yes, yes.
Yeah, so a very good question. So when we talk to manufacturing company, and they obviously have a large set of B. infantis strain that they've been isolated and tested. One of the way that they can distinguish which is the best candidate to move forward for manufacturing is testing their survivability to different environments, including their bile acid response or pH response to what we know the probiotic orally given will pass through the stomach and the different gastric phases. So in terms of how to pick the best candidate, the manufacturing makes sure there will be a high amount of survivability capacity for the strain to be challenged with that environment. So there was no concern to that for B. infantis.
Okay, thank you.
And one of the things, if I may add, is obviously you can look in the feces of the infant of the bacteria itself pre and post administration, and there was totally detected live bacteria within the stool, which obviously it has passed through successfully.
Okay, thank you.